going to talk a little bit about cellular heterogeneity and sort of in the idea about talking more about discussion. I actually don't have any really new data to present to you, but I do have some ideas and some concepts that we might want to think about as we're thinking about the importance of cell heterogeneity in tumors. So in my research group, we work on a lot of different kinds of things, and one of the types of tumors we do a lot of work on is chondrosarcoma, and that's a malignant cartilaginous tumor. And if you look at a histology of a chondrosarcoma, there's all different kinds of cells in it. And my research background is really from developmental biology. So to me, it looks like cartilage development when you think about chondrosarcomas. But I really always had this idea about why is it that all the cells are different? If they theoretically come from a single cell with a mutation, the same mutation, why in many tumors are the cells so different? Some of the tumors are easier to do research on than others. And so a lot of the work we've done recently in cell heterogeneity has been an undifferentiated pleomorphic sarcoma, which is a soft tissue mesenchymal tumor. But the main reason for doing that is that there's some good mouse models that develop tumors really readily, and you can do all these genetically engineered models to be able to look at it. And so several years back, we collaborated with David Kirsch, who used to be at Duke. He's now at the University of Toronto, and he generated a Tyler Jacks, a model in which you could inject in an adenovirus Cree into muscle or into other kinds of tissues in which it would activate a mutant KRAS and deplete P53, and those mice would get these soft tissue sarcomas that phenocopied human undifferentiated pleomorphic sarcoma. So I crossed his mouse with a confetti mouse in which all the cells would be labeled differently. We actually did a number of different labeling techniques. And while we looked at a lot of things about cell heterogeneity, how all these populations behave differently, one of the things that we found that was really interesting is that only a small subpopulation of these tumor cells would go on to form metastases. When they had the primary tumors would become would have all the different kinds of colors, but the metastatic tumors always were the same colors. This would happen even if you took those tumors and transplanted them time and time again into multiple animals, they all were of the same color. And this really showed this concept that within a single tumor, which there are lots and lots of different clones, over time, there would be a dominant clone, but the other clones would still be there. Only a small subpopulation of clones were responsible for things like metastasis. And we had local recurrence, and we tested this by amputating the tumor in the mouse and having the tumors come back in. They were always multiclonal. It really brings up the concept that within an individual tumor, all the different kinds of cells behave very differently, and understanding what's different about those cells, maybe we'll come up with some treatments. Joy Ban, who was a postdoc in the lab, took this on, and she did a CRISPR screen, a functional CRISPR screen, to see what kind of things were different in this metastatic clone than non-metastatic clone. And she found that there were these genes that were involved in an epigenetic complex called the COMPAS complex. And then if she knocked one of the COMPAS complex core genes, the SETD1A, out of these tumors, they wouldn't form metastases anymore in these kind of animals. And that really shows the idea that these metastatic clones may be epigenetically regulated, and that's what makes them behave differently. And if you understand what the differences are and can potentially therapeutically target it, it might have ways that you can sort of treat different kind of tumors differently. And this is from her paper, which was recently published, so you can read it if you want. But it basically shows that the primary tumor, again, is multiclonal. The metastatic cells are always from a single clone, and that if you activate or inactivate the COMPAS complex, if you inactivate the COMPAS complex, which is an epigenetic regulator in the metastatic clones, they won't make METs anymore. And if you functionally activate it, or genetically activate it in the non-metastatic clones, they'll go on to make metastases. And so it shows that there may be lots of things that are regulating different cells that are really important to cell heterogeneity. But you may wonder, is there cell heterogeneity in desmoids? Because a lot of people think about desmoids as being a relatively a less heterogeneous tumor than other types of mesenchymal tumors, or certainly other types of sarcomas. And maybe they all have mutations activating a single signaling pathway in most cases. So is there so much heterogeneity? And years ago, we did some work on trying to look at where tumors that make desmoids come from. And we did some work, just make a guess. There's these cells called pericytes that live outside blood vessels. these cells play a really important role in fibrosis and all different kinds of conditions, like in pulmonary renal fibrosis. And so we generated we generated an animal in which we could see whether or not desmoid tumors developed from these cell types. But we wanted to look at a mouse or an animal that would develop desmoids on their own, and really with Ron Schmitz and Ricardo Fod, who are from the Netherlands, they had generated a mouse, an APC 1638N mouse, which is a mouse that phenocopies attenuated form of attenuated form of familial adenomatous polyposis. These mice get very few GI tumors, but lots and lots of desmoid tumors. And so we basically looked at these and crossed them with a mouse in which we could lineage trace the parasite cells and see whether or not these tumors arrive from parasite cells. And we found that in some of these tumors, they could arise from parasite cells, meaning that that cell was a cell of origin for the desmoid tumors. But very importantly, not all the cells labeled blue. This is a Laxey saying, so not all cells labeled blue. So not all of these cells derive from the parasite. So there's multiple different kinds of cells that are present there. So Mushfiq Al-Jawari, who's was a PhD student in my lab, he's now working at MIT, doing a fair bit of work on trying to understand desmoid tumors. When he was in the lab, he started to look at this about whether or not the different cells in desmoid tumors might behave differently. And he did this by making single cell clones of the desmoid tumors and to see how they behave differently. And one of the things he found, which is probably not surprising at all, is that the desmoid tumors are made of a population of cells with mutations and cells without mutations. So there's these mutant cells inside the tumors, but there's also normal fibrous kinds of cells inside the tumors as well. And he found that the mutant cells would have high levels of activation of Wnt signaling or canonical beta-catenin signaling, and the normal cells would have much lower levels of this. And he found that there were these mutant and non-mutant clones in all the different tumors that he looked at. But one of the things that he really didn't follow up on, which is here in panel C from his work, which I think is really interesting, is that not all the mutant clones had the same level of beta-catenin activation. And there's lots of reasons. He uses axin-2, which is a downstream target in desmoid tumors of beta-catenin mediated signaling. And there's lots of reasons why that could be. It could just be something about the cell culture conditions or something else, but it also could be that even within a single tumor, that those mutant cells may be behaving slightly different. And those differences in how they're behaving could be, like we found their undifferentiated pleomorphic sarcomas, differences in how they're behaving, and it could be potentially therapeutically targeted. He also did look at surface markers that would identify the mutant clones compared to the non-mutant clones, and he found that CD142 is a marker of those mutant clones, and that's actually been shown by the group from Stanford to be a marker for desmoid tumors in a whole variety of tumors that they've looked at, so you can use that. But the cells also expressed different genes. The different clones express different genes at different kind of levels. And you can see that if you looked at the 1, 2, 3, 4, 5, those are 5 different clones that he looked at. This is a heat map of expression of genes. And you can see that even though there were a number of secreted factors that he identified that were made by all these tumors, they were different. So even within the same clones from the same patients, there's differences in what they're making. And that's something that potentially could be looked at to try and answer some of the things that are going on inside desmoid tumors. In the paper he published on this, he was mostly interested in CTHRC1 because that's a secreted factor, and he basically was able to either use it as put a recombinant protein in the cell cultures or use a neutralizing antibody to those. And he could show that he could modestly reduce the proliferation rate in the desmoid tumor cells. And he was suggesting that maybe that interaction between the mutant and the non-mutant cells might be potentially therapeutically targeted. Although I know he's not here, Mushfiq, so I can't really talk for him so much. But I do have to say that the changes saw were relatively modest. So whether or not those are the kinds of things that really could be therapeutically used, I'm not so sure. However, this whole idea about cell heterogeneity within tumors in general, the data that he identified really brings up a lot of interesting ideas about whether or not different clones and different cells within the tumors behave differently? Are there differences in those genes that they're expressing or proteins they're producing? Can they interact with other cells? And could that be some keys into things that we could try to figure out along with thinking about how desmoid tumors behave? So I think some of the areas that would be areas that could potentially could be looked at down the line would be how the different cell types interact with each other. Could it be that recurrence or regression could be related to interactions between cells, or perhaps only certain cell types inside within the desmoid tumors? Could it be that some specific cell types are responsible for recurrence? Maybe not all of them. And maybe this also can do, it could also deal with response to therapy. Maybe some cells respond to some clones within a tumor's response to therapy and some don't. And that could also explain why there might be differences within different patients, depending on what kind of clones are there and how they behave. It also, I think, plays an important role in tumor immune responses, because as tumor immune responses for mesenchymal tumors is already complicated to begin with, because the immune differences, the immune changes, the immune signals are different than in epithelial cancers and other kinds of tumors. But those variations between different clones might have different types of immune responses, which can be responsible for differences in cell behavior. And of course, when you're looking at a lot of variability inside the tumors, it may also play a role in genetic diagnosis. And this idea that some cells have mutations and some don't is one of the explanations for why when you do traditional Sanger sequencing, sometimes you didn't see mutations. It wasn't the mutation wasn't there. It's just that the numbers of cells of the mutation was such a small proportion that it got missed in traditional sequencing. So I think this, to start off the session, this talk is really to get people thinking about how cell heterogeneity might play a role in some of the things we're thinking about desmoid tumors and how people might want to think about doing it. I'm not aware. There may be people who are doing this. I'm just not aware of it, about doing single cell RNA sequencing or the single cell proteomics within the tumors. Those are areas that might show different populations that might be important in this. And again, this work about the individual clones and how they behave differently from Mushrik's work, which really has not been that has not been fully exploited, to why even within the mutant cells from the same tumor, why they behave differently is, I think, something that's really as an area that really could be worked on further. So I'll stop there, and I'm open. I guess I'm open for questions if anyone has questions for me. So you should sure ask them. I can't ask a question of myself. So usually I have a question to ask for me, but other people can ask questions. Yeah. Oh, and everyone should introduce themselves, because I think it's too big a group. When the group was small, we used to go around and introduce each other at the beginning. We're too big for that. But you can introduce yourself when you ask a question. Yeah, go ahead.
When we talk about cellular heterogeneity, that we think about plasticity, and that's one of the things that we want for you. Take a, you look at a single clone or, a cell called plastic, do they work spontaneously only when you use certain factors?
Yeah, that's actually a really good question. So the original reason why I started that work on the undifferentiated pleomorphic sarcoma is we previously had showed that there's like a stem-like cell that is present in those kind of tumors. And I had this idea that there is a stem-like cell that would then make all the other kinds of cells in the tumor. And that stem-like cell would be resistant to chemotherapy and it would stay forever. So originally we did that tracing study to see if that was true. You know what? It's not true. We actually show that's not the case at all. In fact, over time, there is plasticity. Cells can behave differently. However, there's still all those different kinds of clones there. While there's not a stem-like cell that, at least in that mouse model of undifferentiated pleomorphic sarcoma, there's not a stem-like cell that makes all the other kinds of cells, but all those different populations of cells was there at different points in time. And the data on metastases, which we were actually kind of surprised about initially, shows that whether or not there can be plasticity and maybe those cells can change over time, there's still these unique characteristics for those metastatic cells that are really interesting. And we have a Jiro, it's a postdoc in the lab now who's actually looking at that using human undifferentiated pleomorphic sarcomas by doing the same thing Mushrik did by getting individual cell clones from human UPSs and finding the same kind of things, but they vary within individual human tumors, but still the same kind of a concept. So for sure, there's plasticity, and there might even be plasticity in desmoid tumors as they behave, but still if they seem to be the same over time, that plasticity in the interactions can still be used to be able to make treatments. And one of the things that might be important in tumor regression might be interactions. Tumor types might be important in maintaining the tumors. And if one cell population tilts out differently, and then it goes down over time, perhaps that's maybe why the tumors regress. But that's all. It's all. I have no data to prove any of that. Yeah.
The cells cultures were derived from patients. And the second is if you don't think that the cells condition in the culture can play a role in the
Yeah.
association.
Yeah, so there's a good question. In Mushrik's data, these were derived from patients. They were single cells from patients, but the individual patient would have multiple single cells, and they're all in the same culture conditions, but they behave slightly differently. So it's possible that culture conditions could be responsible for why they're behaving differently. Maybe there's something in the cultures that could make them have express a gene differently, but because he kept all the conditions the same, the chance that it's something else about the original clone is more likely than something from the culture conditions. But that's actually really good. I would suspect that if you use different culture conditions, the cells would behave slightly differently. But if you keep everything else the same, if you're really careful about that, and you see differences between the cells, that's likely something intrinsic in the cells rather than from the environment. Tumors, it's always an interaction between the cell itself and the niche that it's in. So those interactions can play important roles, and that's why the important factors of the patients play such a role in how they may respond to tumors.